So this is the green Somso model that depicts the neuron. So neurons can be classified based on their shape. So in this case, this is a multipolar neuron. And neurons are also classified based on the information that they transmit. So they can transmit sensory information and they can transmit motor information. In the case of the neuromuscular junction, for example, that would be a motor neuron. In the case of a peripheral sensory neuron, that would be a sensory neuron. So one is afferent and one is efferent. So afferent is going towards the central nervous system, and efferent is exiting the central nervous system. So you can remember efferent, exiting, leaving the central nervous system. So let's take a look at the microanatomy of this multipolar neuron. So the way the model is arranged, it's not exactly accurate with regards to what you're going to find in the body, in that the neuron has been cut here to accommodate fitting everything on the model. So we'll start in the cell body. So this area of the cell, the neuron, is the cell body. And then there are these little nub structures that are part of the receptive region of the neuron. The cell body is also considered the receptive region of the neuron. And you can see that there are multiple little synapses here onto the multipolar neuron. These are called synaptic bulbs. So this is the synaptic terminal. And these inputs can be either excitatory or inhibitory. And that's going to be based on the type of neurotransmitter that's going to be released onto that neuron. So if we zoom in here at this synaptic bulb, we can see that there are little dots here. Those little dots are going to represent neurotransmitter. So there are a variety of different neurotransmitters that can be released from a presynaptic neuron. So this neuron is presynaptic. It's before the synaptic cleft, which will be a tiny little space here between the presynaptic neuron and the postsynaptic neuron. But they can't include an actual space here because they have to actually attach this accessory to the model. So there are all of these inputs onto this particular neuron, and the neurotransmitter that can be released, it could be glutamate, it could be GABA, it could be serotonin, it could be dopamine, it could be norepinephrine. And each of those neurotransmitters is going to have either an excitatory or an inhibitory effect on the postsynaptic neuron, which is this big multipolar neuron here, specifically at the cell body. So those dendrites are part of the receptive area for the neuron, as well as the cell membrane surrounding the cell body. So in the middle here, we have the nucleus. And inside of that is the nucleolus, and that's going to be where the DNA for the neuron will be found. This is the cytoplasm of the neuron, and these bluish staining structures are Nissl bodies. Nissl bodies. These dark lines, these are neurofilaments. So they are going to be an internal skeleton within the cytoplasm. This structure where we see a wedge shape being formed is going to be very important for the propagation of an action potential, which is the electrical signal that neurons use to communicate with downstream structures, whether it be another neuron, whether it be a muscle, whether it be a gland, the action potential is going to be the signal that's responsible for the neuron's ability to communicate. It is an excitatory cell, and that's what makes it so unique, is that it is excitable, similar to the cells in the heart. Okay, so this structure that's going to be descending from the axon hillock here, which is where the charge will accumulate for an action potential. This is the axon. It is going to traverse away from the cell body. And in myelinated neurons, so not all neurons are myelinated. Thankfully, we have myelin in the human body. If we didn't, we'd be like slugs. And demyelinating diseases can be very pathological. So it's important that our neurons are myelinated. And what myelin is a fatty acid substance that will be laid down in layers that you can see on the model. These individual lamellae or layers of myelin will surround the axon of the neuron, and it will insulate it, except at portions where that insulation does not exist. And this model, even though Somso always does a really good job with their models, is a little bit inaccurate in that they do have a pinching off here of the myelin sheath, but a structure called the node of Ranvier, which is going to be an unmyelinated portion of the axon, is going to be found within these different positions of the myelin sheaths being laid down. So the nodes of Ranvier are going to be very important for the neuron's ability to send electrical signals via their action potentials very quickly in a process called saltatory conduction. And so that is a structure that will be facilitating the ability of myelinated neurons to transmit action potentials at a very high velocity. Unmyelinated neurons are going to be much slower in their synaptic transmission. And as a result, their signals will not reach the brain at the same time as a myelinated neuron. So if you think about when you stub your toe, for example, you know that you've stubbed your toe before the pain actually hits, right? So there's a bit of a delay there in the signal of the pain being transmitted to your brain versus the sensory signal of you having stubbed your toe. And that's because those neurons that transmit that painful stimulus are going to be unmyelinated. So it's not going to transmit as quickly as in the myelinated neuron. And then we have a connective tissue sheath here that will be surrounding the individual neurons. There's a series of 3 of these as you go up in higher levels of organization in the nervous system. And this is the endoneurium that will surround the individual axon within the nervous system. Okay, so back to this myelination. These individual layers are going to be laid down in the lamellae, but there are going to be particular cells that are responsible for laying down that myelination. And those cells are going to differ based on whether or not it's in the central nervous system versus the peripheral nervous system. So in the central nervous system, the cell that's going to be responsible for myelinating the axon is going to be an oligodendrocyte. That is a glial cell. So glial cells are one of the 2 cell types within the central nervous system, but they are not excitable. The neurons are excitable, but glial cells are not excitable. So these are going to be more auxiliary cells
that will facilitate the function within the central nervous system. In this case, the myelination of an axon. So if this neuron were in the brain or the spinal cord, for example, this would be the cell body of an oligodendrocyte. If this was a peripheral nerve going out to your big toe, let's say that you just stubbed, this would be the cell body of a Schwann cell. So peripheral nervous system, Schwann cells are responsible for myelination. Central nervous system, oligodendrocytes are responsible for myelination. And then because there is a lot of activity going on within neurons, it's a very highly energy consumptive cell, our brains specifically, there's going to be a need for lots of ATP. And the cellular organelle, to review, that's responsible for the production of ATP is the mitochondrion, right? The mitochondrion is the powerhouse of the cell. So lots of those here that will be helping the neuron produce energy. And then in this peachish color, we have the axoplasm. So that's the cytoplasm within the axon. All right, so those are the basics of the